Good morning. Welcome everyone to the sixth session of iCEBreak webinar. Today, our invited speaker is Nuno Cerca. Nuno Cerca is principal investigator with habilitation at the University of Minho. He's also associate director of the Center of Biological Engineering at Minho and belongs to the biofilm research group. His talk today is entitled Microbial Interactions in Bacterial Vaginosis. Nuno, thank you very much for your presence. It's a pleasure to have you here. As soon as you want, you can start sharing your presentation. Thank you.
Ricardo, já está a ver? Sim?
Yes.
Ricardo, thank you for the introduction. I'd like also to welcome everyone watching this stream. And today I'm going to talk a bit how bacterial interactions in the human body can lead to disease, in this case, bacterial vaginosis. Before I start, I'd like to make this interesting observation. Within the human body, there is estimates that between 70 to 90% of the number of cells that are within us are in fact not human cells. We have more bacteria in our body than the number of cells that are of human origin. And this observation was the result of a very huge and very significant project called the Human Microbial Project, which analyzed 242 healthy humans, and they detected the different species that exist in our body. And most of these bacterial species, they are not harmful to the human. And in some situations, in fact, they are beneficial to the human host. One of these situations is what occurs in the human vagina. Premenopausal women, healthy women, are normally colonized by a group of bacteria called lactobacillus that you can see here on this image. And these lactobacillus, they are beneficial to the woman because it protects them against infectious diseases. And this is exactly what happens during bacterial vaginosis. So in normal women that are healthy, they have a fair amount of lactobacilli present on the human vagina. But during bacterial vaginosis, these lactobacilli are replaced by a huge number of anaerobic bacteria that you can see here. And you can also see that the human cells, they have a different shape because they are being damaged. This is what happens in terms of microbiology aspect. Clinically speaking, bacterial vaginosis can be described as an abnormal vaginal discharge. And this vaginal discharge is often, they have a fishy smell that can lead to itching or irritation of the vagina. Bacterial vaginosis is the most common bacterial infection on premenopausal women. And as you can see here, it is widespread all around the world. And the two main problems associated with bacterial vaginosis is that if you have BV, like I just explained before, you no longer have the protective lactobacilli bacteria, then you have an increased risk of acquiring sexual transmitted infections, including HIV. Besides this, if you have BV, you also have an increased risk of having pelvic inflammatory disease. So these are the two main problems for women worldwide. However, if you are pregnant or if you are about to be pregnant, then BV brings what I think is more serious problems, not for the woman, but for the baby. This is because if you have BV, there is a chance, there is an increased risk of having preterm labor, or in some situations, miscarriage. So BV, it's a serious disease. It's not life-threatening from the host, for the woman, but it's a serious disease that it's really understudied. And the fact is, after more than 50 years trying to study BV, researchers still do not know what causes BV. So there's very big controversial. And I'd like to point out to these facts here. This data is from 2014. Basically, researchers found that in the most cases of BV, up to 95% cases, there is one bacteria called Gardnerella vaginalis that was present. So they thought maybe this bacteria is causing BV. However, at the same time, the same bacteria is also present on a fair number of healthy women. So this leads to controversial theories. One of these theories presents the idea that it's really just Gardnerella vaginalis that causes BV. And this because there is a very big link to sexual transmitted infections. However, besides the fact that Gardnerella vaginalis not always causes BV, there have been identified more than 300 different species present in BV women. So there is another hypothesis that claims that a group of undefined bacterial consortia working together, they can cause BV. But this is so controversial that some researchers claim that BV is not caused by bacteria. In fact, it's just a genetic predisposition of the host. And this is because Black women have a higher prevalence of acquiring BV, up to 50%. And there are even researchers saying that it's just a physiological condition, something that happens once in a while, not related to bacteria. Despite all this controversial, there are some key research landmarks, research discoveries that over the years led to important breakthroughs on the understanding of bacterial vaginosis. So here I selected a few papers that over a span of 10 years, they start to analyze up to 84 genomes, so the genetic composition of these Gardnerellas. And after 10 years of work involving researchers from different research groups, different countries, they came to the conclusion that what was once thought to be called Gardnerella vaginalis, just one bacterial species, is in fact 13 different species. They are all related, but they are not the same. And with this information in mind, a group from Janet Hill, they realized that women that have BV, the BV symptoms, they have an abundance of two species of Gardnerella, Gardnerella vaginalis and Gardnerella svitinski, but not the other species, which can, if you look at these in a different way, maybe those 40% of Gardnerella species found on healthy women are not the same species that are causing the disease. But if we go back further in time, 10 years back, there was another discovery, a major discovery, done by the group of Alexander Swidinsky in Germany. And Alexander Swidinsky, when he was looking on the microscope to the exudates of vaginal samples, he realized that the Gardnerella that was present on women with BV was in the form of a biofilm. And biofilms, when bacteria are in biofilms, they have very unique characteristics that are not found in normal planktonic bacteria that changes their tolerance to antimicrobials. So this was one of the most important discoveries in the last years that, in fact, led me and my research group to start studying BV. So what is this, the biofilm? I hope that this will work. I have a small animation to show you what happens when a single cell bacteria forms a biofilm. So let's see if this will work. Okay, so a single bacteria will often attach to a surface. In this case, the bacterial, the human vagina. And once they are attached to the surface, they start to grow and multiplicate. And you can see that they will start to produce proteins, DNA, polysaccharides that will create this structure. This is what's called the biofilm. And this structure protects bacteria against antimicrobial agents. So this that are shown here, it's a microbial biofilm. And looking to the perspective of microbial biofilms, there are some open questions that I think they deserve my time and my research group time trying to answer them. And the first question that we tried to understand was, okay, so there are 300 species that were isolated in BV, but are all of these species really pathogenic? Are all of these species doing something? Because we need to differentiate consequence from cause. Maybe we have these 300 species as a cause of BV, is the 300 species causing this disease, or maybe we have 300 species because Gardnerella is able to remove the protective lactobacilli, and in fact, this is a consequence of BV. So one of our first studies was exactly trying to understand that. This image is a bit complex. I will try to make it as simple as possible. We tested 30 different species isolated from Portuguese women with BV, and we characterized two experiments. One was the ability to cause cytotoxicity to the epithelial cells, and the other was the ability to adhere to the vaginal cells. As you can see clearly here, most of these species, the ability to make that initial step, which is adhering to the vaginal epithelium, is very, very, very low. But if you look at the species with the highest ability to be able to start biofilm is, in fact, Gardnerella vaginalis, both on this test and on this test here, which led us to the conclusion that, in fact, not all the 300 species are contributing or have a potential to cause bacterial vaginosis. What's happening here? Okay, so another experiment that we did was exactly trying to understand what happens when lactobacilli disappear. This graphic is also a bit confusing. I'll try to simplify it as much as possible. What is important that I like to show is what happens to these blue bars. And here in this graphic below, we've shown that Gardnerella that were isolated from women with BV, they were able to significantly reduce the protective lactobacilli. But if we are going to use different Gardnerella from healthy women that you can see here, then they were not able to replace the protective lactobacilli that is preventing bacterial vaginosis to develop. Alexander Swidinsky, like I mentioned before, was a pioneer on these studies. It was also interesting to determine the differences between healthy women and women with BV, because most of the studies that I'm talking are, they are not in fact detecting bacteria, they are detecting the DNA from this bacteria. But what Alexander Swidinsky shown here is that yes, women that are healthy, they have Gardnerella. But as you can see here, it's just single cells. But if you have bacterial vaginosis, then Gardnerella is no longer a single cells like you see here, but they form a very thick biofilm. And one of the follow-up studies that my research group did was try to analyze what happens to the bacteria when they shift from a single cell to a biofilm phenotype. And this was the paper that we published. We analyzed it, the 2,000 genes that were being expressed at the same time. And clearly we saw that with the exception of some genes involved on protein export, most of the genes of Gardnerella were being downregulated. Making it simple, Gardnerella in a biofilm is in a less active state. It's almost like it's sleeping, it's hibernating. And in this state, antibiotics normally do not work. So this is a big problem because you try to treat bacterial vaginosis, but once bacteria are in this state, antibiotics tend not to work. Still, we could not neglect the fact that Gardnerella might be important, but maybe there are other species that are also important for bacterial vaginosis. And what you can see on this image here is we try to perform biofilms composed by two different species that are here represented. In pink is Gardnerella, and in blue is other different species. And we find out that many species that are found in BV, they have the ability to increase biofilm formation by Gardnerella, which is represented here in green, which in fact tells me that there are advantages for other species to be associated to this biofilm, and perhaps they will they can increase the difficulty of treating PV biofilms. This advantage that I've been mentioned before, it's a two-way street. Gardnerella gains advantage by some species to be present on the biofilms because the biofilm will be more tolerant to antibiotics, but these are the other species that live there also gain advantages. And in this image that you can see, red is Gardnerella, green is Atopobium, and here is a mixed. We can clearly see that if I grow monospecies Atopobium, after 48 hours, most of the bacteria will eventually die out. These bacteria are very difficult to grow in the laboratory. But if I grow Atopobium together with Gardnerella, then I can maintain cellular viability. This is very important for the field of bacterial vaginosis because it has been shown that the association between Gardnerella and Atopobium is a very important diagnostic marker for the presence of bacterial vaginosis. I'm running low on time, I need to be a bit quicker, but we recently published a new paper where instead of studying biofilms composed by two species, we analyzed biofilms composed of 3 different species that can be represented here in 3 colors, green, pink, and blue. And the important aspect of this study is that not only we are seeing that we can have thicker biofilms, but the expression of some genes of Gardnerella that are involved in virulence can be affected. And with that in mind, our most recent studies have shown what happens when you try to treat biofilms with antibiotics, if they are in a single species or if they are in these triple species biofilms. What you can see here on this image is if I treat Gardnerella biofilms or Prevotella biofilms with the most common antibiotic, we can see that there is a reduction on the biomass of the biofilm. But Atopobium is completely tolerant, so the antibiotic cannot kill this biofilm from Atopobium. So when we grow the 3 species together, you see that the antibiotic is not working anymore. And for those who are experts on this field, they will tell me this is obvious. If the antibiotic cannot kill Atopobium, then after you treat the biofilm with the antibiotic, then Atopobium will grow and all the other species that you can see here, they will die out. But when we observed on microscope this biofilm here, again, you can see the three colors: the blue for Prevotella, the pink from Gardnerella, and green from Microbial. So what this experience shows is that when we have multi-species biofilms, antibiotics stop working as efficient as they work against single species biofilms. And this is the main problem of bacterial vaginosis, because what happens normally, you try to treat with antibiotics, but the treatment fails recurrently. So at this point, and with all this information that I've shown, what we believe is that some species of Gardnerella, they are able to adhere to the vaginal epithelium. They will compete against the lactobacilli that will disappear. And once the lactobacilli disappear, then other species are able to be colonizing this biofilm, and the interactions that can occur between some of these species can cause cytotoxicity to the epithelium cells, producing what is called the clue cells, which is a consequence of the bacterial vaginosis. Main conclusions. Not all species that are present there are in fact relevant for BV, but some can in fact be contributing to the disease. We do not know exactly which of these species are, but these interactions are affecting antimicrobial tolerance, and we need to understand better how these interactions occur, because this will be the only way to develop better antimicrobial treatments. I'd like to thank the research team, the researchers that did work that I presented here today. This is Joana, Aliona, Lucia, Patricia, Daniela, and Angela, and of course the funding from FCT and the Portuguese government that pay all this research. And I'll be taking your questions.
Okay, thank you, Nuno, for this interesting presentation that is within the time that you have. Thank you very much. I can start by a question. So if I understand, a way to track the BV is trying to look for consortia of bacteria in the biofilms, is right? It's not one bacterial species that promotes that, but it's a consortia that can be created in the biofilm.
Ricardo, to be frankly, we are not sure. This is an hypothesis only, okay? What we know, so the process that I'm talking here, it's not something that you can easily do in vivo. You cannot be analyzing women on all these stages. And normally, when you have symptoms of BV, you are already on stage B5. And stage B5 includes a mixture of many species, including Gardnerella. This is the final result. What leads to this final result, it's not known. And this is an hypothesis that we have based on all our experiments and also experiments from other groups. We believe that there is one specific species that can replace the protective lactobacilli that most women have. And once these lactobacilli disappear, then it allows all these other species to be involved there. It is not, I don't think it will ever be possible to know if Gardnerella, I mean, I remember now, there is one paper that they tried to do this in mice, and they find out that Gardnerella itself, even without trying to control the introduction of other species, Gardnerella itself was able to produce the symptoms that we see in BV. Already, this is a laboratory. In real life, you cannot really prevent this to happen.
It's much more complex. Okay, thank you. I think we have a couple of questions also, so I will share. The Slido. I will interrupt your presentation. Where I am at.
Okay, Ricardo, if you can make the question, I'd rather stay with the slides because I might need to use some slides.
Okay, okay. So I will make you the questions. So the first question that I have here is, would phage therapy be a solution to eradicate Gardnerella vaginosis?
So phage therapy, I need to give a broad answer for whoever is listening. Phage therapy is trying to use a specific virus that will kill a specific bacteria. In fact, there are attempts to do this. The problem with, again, treatment comes to this point here. And phage, as a treatment option, will try to kill Gardnerella that is going to be present at this stage. But most likely, because one of the characteristics of phages is they are very specific to a species, and they will not work against these other species. So I think that phage therapy will work perfectly on these stages here, if it could be used as preventive. And like I mentioned in early stages, BV is more of a problem to women that are pregnant. So I will propose that the phage therapy could be used preventively during pregnancy only to prevent these stages to appear. Again, this is a theory, but our theory is if this stage here does not occur, then all these other species won't be able to displace the protective lactobacilli.
Okay, and the next question I think is it's well placed because it's about that too. I think we are talking. Are probiotic cocktails for local application, for example, efficient to treat Gardnerella vaginalis?
Again, this is a challenge to work with this field. Again, there is very controversial papers. Some papers were able to show in vitro, again, all these are in vitro studies, that some probiotics will be able to prevent, by producing lactic acid, will decrease the pH and will prevent the colonization of Gardnerella. But then some attempts of trying to study the application in vivo were unconclusive. In some cases, it will not work. But there is, Ricardo, to try to answer your question, there is a line of research that are trying, because there are many different options. Phages is one, probiotics is other, essential oils from plants is another alternative that we are also trying to do this in our group. So there are many different attempts. However, all of them focus on this stage here. And again, if we do not understand exactly what leads to this stage, one of the consequences of treatment, Ricardo, is this. You can prevent this to happen. So women will stop having BV, but they will still stay on this stage. And once you stop treatment by antibiotics, by phage, by probiotics, you stop the treatment, you no longer have the symptoms, but you wait 3, 4, 5, 6 months, and from this stage, it will develop again to this. So this is a very, very big problem. And I like to share an interesting story. This was the first time that happened to me. A citizen of America, North American woman, non-scientist, she read my papers and she asked me help because she had recurrent PV for years and no doctor on the United States could treat her. It was a very interesting story, and she wanted to see if we could use our essential oil on treatment. And of course, I have to tell her that I'm not a doctor, I'm not a physician, I'm a researcher, and we are long ways to have a proper product that is able to be developed. But for the women that suffer with recurrent BV, it's a very big issue.
Okay. So I think we have also another question. People are focusing in trying to solve the problem. So if it is possible to modulate the vaginal microbiome to deal with the bacterial vaginosis in a personalized manner. I think it's
in the personalized manner, I'm not exactly sure what that means, but trying to modulate the vaginal environment, that's what's been trying to do with the probiotics. The lactobacilli here, they maintain the pH of the vagina acid. Once they disappear, the pH raises, and then this can happen. We do not know what leads to the disappearing of lactobacilli. Our own experiments shown that in the laboratory, again, this is not human experiments, in the laboratory, Gardnerella is able to promote the displacement, but it can be other reasons. Like I mentioned before, some scientists believe this is a physiological response, can be a physiological response to stress, for instance, to the hormonal flow. So there are many possible reasons why this is occurring. So the way to modulate the vaginal environment is trying to introduce lactobacilli and trying to maintain pH low. And to some extent, it's successful, but it does not prevent all the cases of PV.
Okay. And you see any upcoming challenge from a point of view that will be worth to study, or kind of next steps?
The upcoming challenge it's universal to all bacterial infections. We are living in an era of antimicrobial resistance. There are more and more bacterial species that are becoming resistant to antibiotics. And in some situations, this can lead to the person death. Like I said, bacterial vaginosis does not cause the death of the woman. It can cause preterm labor or a miscarriage. But with this increased level of antimicrobial resistance, the cases of untreated BV will become more and more. So I think it's very, very important trying to find better options of cure. And I think that in order for us to not only treat the symptoms, but treat the infection, we really need to understand the fundamental of what's happening between these species, the interactions between these species.
Okay, thank you. I think we have one more time for one more question. If the bacterial vaginosis can be diagnosed in a clinical and rapid way?
Not a problem. There are two options of treatment. The most common is called the Amstel criteria, and basically the doctor will try to analyze some symptoms, like, for instance, if the woman has the vaginal discharge, if it has the fishy smell, and also, what is the other one? Oh, and looking at the microscope, if you can see, I can go back to these images here. If you can see the clue cells, where they are, these ones. So these are part of the clinical diagnosis. It's quick, but it's not very sensitive. So only about 60 to 80% of the cases can be diagnosed like this. And there is another problem. Some of the cases are misdiagnosis. There are now attempts, Ricardo, to make molecular studies.
We have to close the session. Thank you very much, and
thank you for everyone.
We see you in September again, and have a nice holidays for everyone.
Bye bye.
Okay, bye bye.
Boas férias.